All right, everybody. Good afternoon, good morning, good evening. I hope everyone is well. This is the last supplemental video for muscle tissue in chapter 10. So we're going to finish up. As you recall, there are three types of muscle tissue. We spent a majority of the time talking about skeletal muscle tissue, but now we're going to talk about cardiac muscle tissue and smooth muscle tissue. Other chapters in the course. For 210 and 211. We'll go into more detail about the latter two muscle tissues, but we're going to kind of cover some of the basics. Some of this you already know from chapter 5 when we were talking about the tissue types. Beginning here with cardiac muscle cells. We know that they're shorter. They're not going to be long and cylindrical like the skeletal muscle tissue. Plus, they branch. I often tell folks, it's like taking the letter Y and laying it on its side. And that's what we're going to see when we're looking at cardiac muscle tissue, this nice branching kind of configuration. These cells can have more than one nuclei. It's either one or two, right? And they share a similar quality with skeletal muscle tissue in which they're striated. And that's because we know that with the striations is due to the configuration of the sarcomeres. So yes, cardiac muscle cells do have the sarcomeres. And as you know, your cardiac muscle tissue is quite busy. Quite busy because obviously when you're sleeping, it's still pumping, it's still working. So it requires a lot of energy. Well, if you require a lot of energy, then you require a lot of mitochondria because primarily these muscle cells use aerobic cellular respiration. But there is something new with cardiac muscle cells, and those are those intercalated discs. I've mentioned this before. And the intercalated discs are going to be found in between the muscle cells. And they're wonderful structures. You'll talk more about it when you get into the actual cardiac chapter. But these intercalated discs are going to be made up of desmosomes and gap junctions. Right? One of those structures helps to hold the muscle cells snugly with one another. It actually makes contraction more effective. And the other one is going to essentially create these holes in between the cells, and it allows the ions to flow from one cell to the next. They experience less resistance. And as you know, when we're trying to depolarize these cells, right, resistance plays a role in that. Well, the gap junctions help to decrease that role of resistance so we can depolarize and actually get these cells to contract. So again, you'll get into more detail on all that in later chapters. All right. Also, we have specialized cells in our cardiac muscle tissue, which we call pacemaker cells. And the pacemaker cells are going to be the cells that are going to trigger the cardiac muscle cells to contract, but they're going to kind of, like the name implies, they're going to pace the heart, right? We have an automatic heartbeat in which without nervous system stimulation, your heart can still beat on its own. Unfortunately, it beats quite fast, and it's just too much. And so the nervous system helps to kind of slow that down. There's a movie that came out in the 80s called Indiana Jones and the Temple of Doom, and this always makes me think about it. One of the villains of the movie had this ability to reach into people's chests and pull out their heart, and the heart would still be beating. And so that is actually true, that the heart will beat because of these pacemaker cells and their effect on the cardiac muscle cells. Keep in mind, when I say that the cardiac muscle cells are under the influence of the nervous system, specifically our autonomic nervous system. All right, the autonomic nervous system is going to be that other part of our nervous system. We have the somatic nervous system. That's the nervous system that we're consciously aware of stimuli, and that nervous system, the motor aspect of it, is the one on which we can consciously and voluntarily control. Well, the autonomic nervous system is like our body's autopilot. And so the cardiac muscle cells are under that influence there. All right, here's a picture of our cardiac muscle cell. You can see how it branches. There's a couple of nuclei there. The cell itself is littered, chock full of mitochondria, right? And we do have our sarcomeres. And we'll see, if we zoom in here a little bit, you can see that you have your gap junctions. Here are the gap junctions. They're these proteins, 6 proteins that create a little hole in between the cells so ions can flow from one cell to the next, very effective in reducing resistance. And then our desmosomes are almost like someone took a thread and needle and just kind of sewed the cells together tightly. And that's what it does. It holds these cells, tight against one another so we get into a more effective contraction, which is quite important when we're dealing with cardiac tissue. What are some of the differences between skeletal muscle and cardiac muscle? Here we go. Here's all our cardiac muscle cell characteristics, 1 or 2 nuclei, branching cells, short, thicker cells. We do have intercalated discs here, and these cells are going to be stimulated by our pacemaker, our autorhythmic pacemaker. Whereas the skeletal muscle tissue, okay, can be multinucleated. Well, it's not that they can, they are multinucleated, all right? They're long, cylindrical, non-branching cells, no intercalated discs, all right? And yet cardiac muscle tissue will be stimulated by the nervous system, or our somatic nervous system. Okay, last but not least, our smooth muscle tissue. All right, now if you look here on this slide, you can see all the different places that smooth muscle is found throughout the body. And as you go through each of these different systems here, and unfortunately we don't cover a lot of these systems this semester, you'll cover them in 211, but look, cardiovascular, respiratory system, digestive system, urinary system, all right, in the reproductive system and other places, you find smooth muscle. Smooth muscle is all over the place. So you can see that the functions of smooth muscle, yes, are going to be to move things. Keep in mind, so obviously in the cardiovascular system, it's going to help regulate your blood pressure. It can cause vasoconstriction or vasodilation, make the blood vessels bigger or smaller, the lumen of the blood vessels bigger or smaller. Just like more water flows through a larger pipe than it does a smaller pipe. That's what we'll see here in the cardiovascular system. Respiratory system, similar type of concept. The breathing airways there can become bigger and smaller. All right, we call that bronchoconstriction, bronchodilation. So that affects the airflow, all right, of the air that is leaving your body or entering your body into and out of your lungs and the alveoli there. Digestive system. Think of the digestive system as one long tube. Once we get it to the stomach, whatever you've ingested, liquid or solids, right, it's going to move from the stomach down towards the anus. Well, what helps with that movement is going to be the smooth muscle tissue, right, moving it towards the exit of your body. Right? So as you go through this course, you're going to see, right, the role of smooth muscle, and it's vitally important. Keep in mind, smooth muscle is just that. It's smooth. No striations, no sarcomeres. And, all right, it is under, all right, the nervous system control that we do not consciously control. So that's going to be your autonomic nervous system. All right, so you can see this picture here is showing you all the vast and different places that smooth muscle is located throughout the body. So let's talk about a smooth muscle cell. I think that it looks kind of like a football. All right, we call it a fusiform shape, similar to fibroblasts. It's thick in the center, right, and tapers at the ends. That allows for these muscle cells to be tightly packed into one another, right? So we can get cram as much in there, all right, as possible. Now, what we'll see that's different with smooth muscle cells compared to cardiac and skeletal is that we have a centrally located nucleus and we are missing our sarcomeres. But one thing that we'll notice on the plasma membrane, the sarcolemma, we have, all right, different types of calcium channels. Some are chemically gated, some are voltage gated. So it depends, there's just different channels depending on what type of stimulus that we're going to be utilizing. Another quality that we see in smooth muscle cells is there's no T-tubules, but instead we have a different structure called a calveolae. And instead of it looking like a T-tubule that just pretty much drops down like this, these are flask-like invaginations, which look more like this. Right, now you might be thinking, there's not much of a difference there. Believe you me, there is a difference, okay? So no T-tubules, but the calveoli are present. And also, yes, we have sarcoplasmic reticulum, but it's not going to be as abundant in our smooth muscle cells. So as you recall, the sarcoplasmic reticulum is where we keep our calcium. Yay, we love calcium. We need calcium for muscle contractions. But unfortunately, all right, since it is sparse, we have to rely on getting calcium elsewhere. Well, where is that elsewhere? That elsewhere is outside of the cell, right, in the interstitial space. So more on that in a little bit. So here's a nice picture here of our smooth muscle cells. Okay, some of the things that you're noticing, here's our centrally located nucleus. We have some mitochondria, All right, it's a fusiform shape, almost like a football here, tapered ends, thicker in the center. You can see the calveoli here, all right, these invaginations into the cell. All right, now, as we kind of zoom in here and look closer, you can see the difference between a relaxed smooth muscle cell and a contracted smooth muscle cell. A relaxed smooth muscle cell here is on the left. You can see how they're tightly packed in against one another. And then you can see what happens when they contract here. Now we're missing our sarcomeres, but we still do have, all right, our myofilaments of actin and myosin. And so you can see they're arranged in these triangular shaped configurations here. So when they're relaxed, right, there's some overlap that's occurring. And the actin filaments are anchored to these structures called the dense bodies. We'll get to that in a second, okay? But if you notice, when they're relaxed, there's a little bit of overlap between the thick and thin filaments. During contraction, there's quite a bit of overlap, which you would assume, right? But you notice that you have these three dense bodies in each of these pictures. Look at the spacing in the relaxed muscle. Look at the spacing in the contracted muscle. They pull closer together. And so that's what we'll see here with smooth muscle. We'll get into this in a moment, right? But I want to just draw that to your attention here as we move through the remaining material of this chapter here. All right, so let us talk about how we've arranged, all right, our thick and thin contractile proteins there, our actin and myosin. So in this cell, our cytoskeleton, we will see, all right, intermediate filaments all over the place. And so these intermediate filaments, right, what we call dense bodies, which I just talked about here, and these dense plaques here, you're going to see how whereas with the dense bodies, all right, these dense bodies are going to interact with our sarcoplasm, whereas the dense plaques are going to interact, all right, with the sarcolemma. So those contractile proteins, those thick and thin, all right, filaments, all right, are going to be found in between our dense bodies and the dense plaques. So you're going to see, and that's why I don't know why it reminds me of that, but when you look at the smooth muscle cell here on the right contracting, I always think if you've ever gone to the butcher's, I don't know what kind of meat it is, I'm sure it's various types of meat. Sometimes they have this string that covers, like if you buy a roll of this meat and the string's pulled tight, it digs into the meat. That's what these contracted smooth muscle cells look like. But what we're seeing is when those smooth muscle, the myofilaments are contracting and the distance is shortening, right? It will pull those dense bodies closer to one another. And those dense bodies will have these intermediate filaments attached to it. But these intermediate filaments will also attach onto the sarcolemma. That's the plasma membrane. And it'll attach onto the sarcolemma, all right, to the dense plaques. And so that's how we get that overall, all right, contracting here of the smooth muscle cell. So that's what this picture is kind of showing you here. All right, here are your dense plaques littered throughout the sarcolemma. And then once the muscle contracts, right, it pulls on those intermediate filaments and it will cause that the dense plaques, right, to move closer towards the center of the cell. Okay, so when we see contraction here of the smooth muscle cells, think of it like a twisting motion, like you're wringing out a washcloth. That's what happens. All right, so I mentioned before, no sarcomeres, no and Z discs, so we have no striations. So let's talk about a little bit about how, all right, this tissue is going to contract. Something that you need to understand first is what is present in a smooth muscle cell. So in our smooth muscle cell, we are going to have our contractile filaments, myofilaments, actin and myosin, right? We'll also have that tropomyosin, but notice we do not have troponin. No troponin is present in our smooth muscle cell, right? So what we're going to see here, when our actin and myosin filaments interact, we will get cross bridge. All right, so we already know, familiar with that process of what occurs with our cross bridge mechanism. But we have a new concept now when we're dealing with smooth muscle. We have this concept called the latch bridge mechanism. And so this is a nice thing, because with our bridge mechanism, right, our myosin and actin cross bridging can stay connected for a longer period of time without using extra ATP. So it almost kind of locks in place for a short period of time, which is beneficial, right, when we're trying to deal with the contractile process of smooth muscle. So the fact that once they've created that cross bridge and they've locked into place, right, we can have that contraction occur over a significant period of time without using extra energy. That's wonderful. Now, you may have heard of this protein here called calmodulin. All right, if you remember it in the terminal cisterna in skeletal muscle tissue, all right, there was two storage proteins. There was calmodulin and calsequestrin. So they'll bind onto protein. So calmodulin in smooth muscle will bind onto calcium, and this is going to trigger the contraction. So kind of a different process here. I'll show you here in a picture that I have in the next slide here, okay? But the calmodulin, pretty much the same role in which that it binds to calcium, but in this case, once it does, it's going to trigger the contraction. So we have two other players involved here in our smooth muscle contraction. One is called the myosin light chain kinase. All right, we know it's an enzyme because it has ASE on the end. And it's an enzyme that likes to stick phosphate onto something, okay? We call that process phosphorylation. So what will happen is this enzyme is going to phosphorylate, stick a phosphate onto the myosin head there. Once calmodulin triggers this to occur. Then, like I've always said, what goes up must come down. If we want to stop this process, we bring in another enzyme. It's called myosin light chain phosphatase. And this enzyme will take that phosphate off the myosin head, and that will trigger relaxation. All right. Like always, I like to bring in a picture. Let's take a peek. All right, so let's start off up here at the top. We got our calcium outside the cell, because remember what I said there, all right? We don't have a lot of sarcoplasmic reticulum, okay? So it's sparse. So we need to rely on getting calcium elsewhere. So that calcium is outside of the cell in this example here. So what will happen is, all right, the autonomic nervous system is going to trigger the opening of these voltage-gated calcium channels through an action potential. And so when that happens, we know the story, calcium enters into the cell. And what will happen is calcium will bind on to this calcium binding protein here, the calmodulin, and that's going to create a conformational state change. It changes the shape of it. And so when the calcium binds on, it changes the shape of this protein. And then what happens is this protein and calcium complex are going to activate the myosin light chain kinase. Once activated, what it'll do is it's going to add, all right, phosphate to the myosin head here. And that will then cause, all right, the interaction of myosin with actin. So we're going to get that cross bridge formation. Once that occurs, all right, that cross bridge formation, we'll get, the same story, the power stroke. And then we'll release from the power stroke, all right, and then release the cross bridge and maybe undergo another contraction. Or we can sustain the contraction through that latch bridge mechanism. So that's good. This is a little bit of a different process. I love this picture because it kind of walks you through the steps here. And so that's what we're going to see, all right? We see that conformational change here with calmodulin, which activates this enzyme, the myosin light chain kinase. That myosin light chain kinase rolls in, and it will then phosphorylate, all right, the myosin head. And then what we'll see now, once that myosin head is activated, it will then create that cross bridge formation with the actin. And this will continue on as long as we have ATP present. Now, when we want to inactivate, all right, our myosin light chain or inactivate the myosin head, what do? We get our myosin light chain phosphatase come in, and it will dephosphorylate the myosin head, and we can stop the process there. One of the nice characteristics of smooth muscle is that we consider it to be fatigue resistant. And that's partially through, all right, that latch bridge mechanism. All right, so again, energy requirements, low. But we are still going to get what energy we do need through aerobic cellular respiration, which gives us plenty of ATP. And so this will allow this smooth muscle to contract for an extended period of time without tiring out. We learned about what fatigue is, muscle fatigue, at the end of a contraction, at least sustained contraction. This is what makes smooth muscle so useful throughout many of our body's systems. Think about the stomach. I mean, after you eat, right, the stomach, think of it, it's like a cement mixer. It's going to be spinning and churning and trying to mix up everything that you've ingested, liquid, solid, and then any of the secretions that the stomach is producing and some of the secretions that came from the oral cavity. It's going to mix all that up, and it'll be constantly doing that, helping to try to break down the contents, but mixing in the enzymes, the digestive enzymes that you need for proper absorption of these nutrients. You get to learn about that. I believe it's chapter 26, fun stuff in 211. See, there's so much to look forward to in 211. And then finally, of course, this type of muscle tissue is going to be innervated by, all right, our autonomic nervous system, basically conferring that it is involuntary. You do not have conscious control over smooth muscle contraction. All right, folks, that's the end. This is a short one, okay, but that is the end of chapter 10, muscle tissue. I hope you enjoyed this video, and I look forward to giving many more. Have a great day.